So our first abstract for the morning is going to be training autologous tumor-specific T cells for effective immunotherapy of neuroblastoma. It is going to be presented by Dr. Sarah Wright.
Hi. Good morning. Thank you for the opportunity to present our work. I'll be discussing a novel method to train autologous tumor-specific T cells for the treatment of neuroblastoma. I'm a military resident, and these are not the views of the Department of Defense. Despite advances in conventional treatment and immunotherapy, neuroblastoma remains challenging to treat, especially for advanced stage, recurrent, and refractory tumors, where survival rates are as low as 10% for these populations. Adoptive immunotherapy, which involves transferring T cells and natural killer cells to patients to specifically target and eliminate cancer, has had promising results in other tumors, but there's still a lot of work to be done in neuroblastoma. This research is important because it introduces a novel method of training tumor-specific T cells that exhibit potent killing of human neuroblastoma, and we think that this method of adoptive immunotherapy conveys some advantages to the standard approaches. Current adoptive immunotherapies focus on ramping up the immune system through the addition of a chimeric antigen receptor or a specific T cell receptor, or by using a small population of targeted cells that are known to target the tumor. But all of these strategies are trying to overcome cancer's innate defenses and the pathways that the cancer uses to evade recognition and elimination by the immune system. Immunosuppression due to MYC-N amplification is significant in neuroblastoma, and it's one of the strongest predictors of aggressive disease and poor prognosis. Neuroblastoma also expresses PD-L1 and has a hostile microenvironment, which is a hallmark of solid tumors that makes it especially difficult to treat with immunotherapy. So to address these challenges, our approach to training T cells starts by taking isolated tumor cells in vitro and breaking down these defenses so that the tumor is easier to target and is more immunogenic. We have primary cultures of human neuroblastoma developed from tumor specimens obtained from 8 patients at Children's National. 2 of these were MYC-N amplified, all were intermediate or high risk, and these primary cultures express FOX2B and synaptophysin, which are markers of neuroblastoma. When we treat these tumor cells with small molecules BET and JQ1 that are inhibitors of MYC transcription, we see morphologic changes in the cells and also suppression of MYC-N, c-MYC, and PD-L1. To assess the impact that MYC downregulation has on immune function, we took PBMCs, so containing B cells, T cells, NK cells, monocytes, and dendritic cells, and co-cultured them with irradiated tumor cells that were treated with these MYC inhibitors, BET and JQ1. The co-culture of the BET and JQ1 treated tumor cells resulted in significantly higher levels of interferon gamma and granzyme B compared with co-culture with wild-type tumor, and showed a significant upregulation in several pro-inflammatory cytokines, showing that MYC downregulation with these small molecules induces a tumor that is more susceptible to a substantial immune response and is more immunogenic. So when these PBMC are expanded, they're greater than 90% CD3 positive by flow cytometry, and the CD8 population also of effector T cells is increased in the PBMCs that were co-cultured with BET and JQ1 treated tumor cells. When we looked closer at the treated tumor cells themselves, we found another immune pathway that was implicated, STING. And after treating tumor cells with inhibitors and activators of STING, we found that the STING inhibitor C170 boosted production of interferon gamma in co-culture with PBMC, and so we added it to our training protocol to induce immunogenicity in these tumors. So from that work, this is what the training now looks like. We take patient PBMCs and isolated tumor cells, irradiate them and treat them with BET, JQ1, and C170 to create this immunogenic tumor, and then co-culture them for 7 days of training to create our trained autologous tumor-specific T cell product, or TATS-T. And then to assess the function of these TATS-T, they're co-cultured back with the wild-type primary tumor cells, and we want to see how well they kill. So what we find is that there's increased levels of interferon gamma, caspase-3 and 7, and granzyme B when these tumors are trained, or when these T cells are trained with the immunogenic tumors and tested against wild-type. One of the assays we use is an incucyte killing assay. It shows T cell killing in real time. Tumor cells are marked with red, and then caspase 3 and 7, which are markers of apoptosis, are seen as green fluorescence. And when the software puts together these two dyes, you get explosions of yellow that show when T cell killing is happening. And we see that the most robust T cell killing happens when we're using PBMCs that have been trained on BET-JQ1 and C170. Here we're looking at neuroblastoma in red with co-cultured with untrained PBMC. And then when we co-culture with our TATS-T trained T cells, we can see that the T cells track, smother, and kill the tumor cells. This research is important because it shows that treating tumor cells with MYC inhibitors makes the cells more immunogenic and more susceptible to targeting by immune cells, and we think this method of adoptive immunotherapy can induce a robust T cell response against the patient's own tumor. This research is the product of many years of work by Dr. Xiaofeng Wu and Masumi Basu in Dr. Sandler's lab at Children's National, and I'll take any questions. Thank you.
Hi, Lindsay Talbot, St. Jude. That was really great. I love the Inky site as well, and we love that caspase dye. I want to ask you sort of a theoretical question. Some of us in the field are coming to understand that perhaps the way to have T cells have long-term efficacy in vivo is to actually make them more stealth T cells, where they're not producing all of these cytokines and they're not attracting in other immune cells from the endogenous immune system during killing, so that immunosuppression doesn't happen. Have you tried this approach in vivo, and do you see sustained killing in vivo models?
We haven't tried this approach in vivo. I don't know if I still have slides or not, but it's okay. We haven't tried it in mouse model, and it does show effective killing. I think our model, we're working on making a more intense model, more challenging model for it, but it does show good killing and not very much autoimmunity. So looking at like cardiac cells, the autoimmune profile and the inflammatory profiles of those mice are not showing the kinds of autoimmune or like cytokine release issues that we see. I think for this therapy, it has some benefits over some of the other adoptive immunotherapies because it doesn't have the co-stimulatory domains that you worry about with CAR that like make the immune response more ramped up. This is it's a normal immune system that has just been taught and shown and exposed to the antigens that it needs to be exposed to in a way that's not as artificial as some of the other. So I think it's one of the benefits from it.
Do you see microenvironmental change? You said you are trying it in mice. Are you evaluating the microenvironment, and do you see that your hypothesis reflected in that?
We have not done as many, we haven't done the experiments for microenvironment in the in vivo model, but I think that the in vitro is not really a good, as you know, representation of that. But I think the fact that we're getting killing in vivo shows that it's at least able to overcome the basic microenvironment that's there. Sure. But yeah, definitely more work to be done.
That's fascinating. I'd be really interested to hear more.
Thank you.
Beautiful work, thank you.
Thanks.
I'm Joel Shalansky from University of Iowa. I have two simple questions. One is one of the challenges for adoptive immunotherapy is expanding. I'm sorry. One of the challenges with adoptive immunotherapy is expanding your cytotoxic T cells.
Yeah.
So how well are you able to do that? Because you only culture them for a week, I think. And then the other question is, your model suggests that something about the tumor cells inhibit either the presentation or activation of cytotoxic T cells. And so inhibiting MYC appears to obliterate that inhibition. And have you looked at the mechanisms by which the tumor inhibits T cells or T cell activation?
Okay, so the first question about proliferation and expansion of T cells. The BET and JQ1 small molecules do have like a bit of an inhibitory effect on growth. And yeah, it is a challenge to grow these cells up. I think some of the benefits that it has is that we can use the whole population of PBMC, and we see that the 30% lymphocyte that we get in that is sustained, and we can use all of those cells as opposed to having to transduce or find T cells that are coming from like TILs that are coming from in the tumor. So it's going to be a challenge moving to an IND and GMP work, but I think we have some benefit. Right now, we've been doing everything with peripheral blood, and I think we'll be able to do that in the clinic a little bit, but ultimately, leukapheresis product is much better for you. And then the second question of why MYC inhibition is allowing for this. There's some previous work in our lab showing that MYC inhibition has implications for antigen presentation and processing, and especially with STING inhibition also, we see downregulation of DNA repair pathways in a way that allows for more antigen presentation, which exposes more things to the immune system so that more T cells can have a target to go to. I think the fact that we don't know exactly what the target is right now, we're working on it. There's probably, the work that we're doing with a collaborator in New York shows that there's about 70-ish clones that it could be, and or a mixture of that. And I think that's an advantage too. We're not getting antigen escape if you're targeting many things.
Mm-hmm. Thank you. That's very good work. Thanks.
Thank you so much. Thank you.
Our next speaker will be presenting Identifying Novel Genetic Markers in Pediatric Rhabdomyosarcoma. Welcome Dr. Xu to the podium.
Hi everyone, good morning. My name is Joyce. I'm a third-year medical student at Rutgers Robert Wood Johnson Medical School, and today I'll be sharing my project, Identifying Novel Genetic Markers in Pediatric Rhabdomyosarcoma. So Dr. Bertrand Rapa and I were discussing a patient with rhabdomyosarcoma, and I had raised the question of why is FOXO1 fusion status the only genetic marker that is incorporated into risk stratification? Thus, we decided to start this project to investigate if there's any novel genetic markers that are associated with prognostic implication and could be used for targeted drug therapies in rhabdomyosarcoma. I have no disclosures to declare. So some background first. Rhabdomyosarcoma is the most common soft tissue sarcoma in children, with approximately 350 new cases in the US each year. Prognosis of low-risk rhabdomyosarcoma has improved greatly in recent years, but prognosis of high-risk sarcomas remains poor. Ongoing clinical trials are being conducted by the Children's Oncology Group, and risk stratification has traditionally been determined based on tumor histology and staging, and only recently has molecular features such as FOXO1 fusion status been incorporated into the risk stratification. Thus, our aim was to identify novel age-specific genetic markers associated with prognostic prediction in pediatric rhabdomyosarcoma. We started with 641 patients with rhabdomyosarcoma in the NCI Oncogenomics Clinomics dataset. We included patients with at least one gene mutation of interest and excluded patients with unknown risk stratification or unknown age of onset. We identified mutations of interest as genes with at least 5% mutation incidence in the initial dataset. This included 3 tumor suppressor genes, BCOR, NF1, and TP53, 3 RAS signaling proteins, NRAS, HRAS, KRAS, and 2 other signaling proteins, FGFR4 and CTNNB1. Our final cohort consisted of 370 patients with these mutations, which we further stratified based on age of onset. There were 51 patients in the 0 to 2 years age group, 140 patients in the 2 to 5 years age group, 112 patients in the 5 to 12 age group, and 67 patients in the 12-plus age group. Notably, less than 6% of these patients were fusion positive. Our first finding was that in patients with greater than 1 mutation of interest, later age of onset is associated with poor prognosis. When we look at the 12-plus age group, we see that event-free survival and overall survival are about 3 to 4 years, which is significantly poorer than all of the younger age groups, which have an event-free survival and overall survival of 5 to 6 years. In patients with only BCOR mutations, we see that later age of onset is also associated with poor prognosis, where the event-free survival and overall survival in all of the age groups greater than 2 years is about 4 to 6 years, compared to the 0 to 2 age group, which has an event-free survival and overall survival of about 11 years. In patients with only NF1 mutations, we also see that later age of onset is associated with poor prognosis. And in the age groups that are greater than 2 years, we see an event-free survival and overall survival of about 4 to 6 years, and that is also significantly poorer than the event-free survival and overall survival in the 0 to 2 years age group, which is about 9 to 10 years. So in conclusion, we identified 8 genetic mutations that have greater than 5% mutation incidence in our initial data set. This included tumor suppressor genes, RAS signaling genes, as well as miscellaneous other signaling genes. Notably, less than 6% of these tumors were fusion positive, making the carcinogenesis or ways that we could target it separate from the fusion positive tumors. We saw that the tumor suppressor genes and the RAS signaling genes had the greatest incidence in this data set, and we also identified BCOR and NF1 as promising age-specific prognostic markers that could be targeted with future therapies. So these findings definitely do need to be validated in larger data sets, but we hope that our findings would be able to be incorporated into targeted therapies for patients with genetic susceptibilities. For example, we could, in patients with BCOR mutations, they have aberrations in epigenetics, so we could use epigenetic modifiers or targets to target those specific populations. Additionally, in patients with RAS signaling mutations or mutations in NF1, which is a negative regulator of RAS signaling pathway, we could target the downstream pathway with MEK1 inhibitors. So thank you, and I'm happy to take any questions.
Hi, Rashida Skepta from Cincinnati. Thank you. That was really interesting work. I just don't know a lot about this NCI oncogenomics data set. Are these patients that are known for sure to have rhabdomyosarcoma? Because BCOR, as you may know, has now been a separate entity in terms of sarcoma based on the CCNB3 mutation. So it's a completely different entity other than rhabdomyosarcoma. So I wonder if some of the patients that were initially diagnosed as rhabdomyosarcoma aren't really rhabdomyosarcoma as some of these new mutations have been sort of figured out. Do you know anything more about that?
Yeah, so when we looked at the original data set, genomic classification as well as tumor histology was all done for these tumors, and they are all confirmed to be rhabdomyosarcoma, mostly alveolar and embryonal subtypes.
Nice presentation. Tim Laus from Lurie Children's. Excuse me. As you think about your conclusions, I would encourage you to think about not only potential targeted therapies, but also risk stratification. Because a lot of the movement now has been to understand even the patients who would fall into low versus intermediate risk, using molecular markers to really identify which patients can have therapy de-escalated and which ones need intensified therapy. So that could potentially be an avenue for taking your results forward.
Thank you.
So if I could thank you, a question. So I was really surprised that the fusion positive status was only less than 6% of your patient population. So I'd like for you just to comment upon that. The second thing is rhabdo is notorious for having a low frequency of mutations. And so doing this kind of targeted therapy can be difficult because it's such a small part of the population. Those are just two observations. But two questions would be, did you try seeing if these mutations were additive in their effect? Did you try combining them to see if multiple mutations even gave a worse kind of prognosis? And then the second thing is age in and of itself, greater than 10, is a poor prognostic factor and has a poor outcome. So is what you are seeing, is it age-related? Is it really the mutation-related? Or is the mutations the explanation for why age greater than 10 does worse? Thank you.
All right. I might have you repeat the questions one by one, but the first question,
Sure.
regarding, I got the first one regarding fusion status. So just commenting on why it's less than 6% in my data subset. In the initial data with the initial data with 641 patients, the incidence of fusion positive tumors was about 16%. But we had further excluded, or we only included tumors with mutations of interest, and that group had 340 patients. So within patients with those 8 mutations, only 6% were fusion positive.
Okay. So the next question is the effect of the mutations additive.
We were not able to perform that analysis. On average, each of the tumors that we saw only had 1 mutation, or at most 2 of these mutations of interest. And in our smallest group, there were only 5 patients with a B-core mutation, for example. So statistically, it wasn't really possible to perform that analysis.
And then last was age versus mutation.
Age versus mutation. So when we looked at B-core mutations only and NF1 mutations only, the biggest divide was between ages 0 to 2 versus ages 2 and older. I know that age greater than 10 has been something that's incorporated into risk stratification because it's known that patients greater than 10 have worse prognosis, but I would have expected the divide to be patients less than 10 versus patients older than 10 in this data set if that was a confounding variable. I'm sure it could play a factor, but I think that the patients ages 2 through 10 also seem to have poor prognosis if they have these B-core or NF1 mutations.
Rita Burchiga, Dartmouth. Congratulations as a medical student to be up here answering to the national experts over rhabdomyosarcoma, 1. 2, I saw NF1 and P53. Do you know, are your patients having also some germline mutations? Do you know their germline basis or not in your data set?
That's a great question. That's something we posed through our data analysis process as well. Unfortunately, we didn't have the data available for germline versus somatic.
Thank you.
As a third-year student, well done.
Thank you.
All right, our third abstract is Social Determinants of Health Impact Neuroblastoma Progression in Vivo, and is going to be presented by Dr. Kiona Williams.
Good morning, and thank you for this opportunity to present. Neuroblastoma is one of the more common pediatric cancers, accounting for about 10% of the pediatric cancer deaths. And we know that despite multiple therapies, patients with high-risk neuroblastoma have less than a 40% chance at 5-year survival. Social determinants of health is a new topic that has been trending ever since COVID-19 pandemic. And the World Health Organization and CDC defined it as non-medical factors that affect health outcomes. These health outcomes can range from where a patient lives to where they're born, and they can wider ranges of systems that affect their daily conditions of living. We did this research to show that we can create an animal model to study the effects of social deprivation, and we wanted to present these findings from our pilot studies and show how they specifically affect neuroblastoma pathogenesis. Putting social determinants of health and neuroblastoma together, the Children's Oncology Group did a study that found that poverty was independently associated with the increased risk of relapse and death in high-risk neuroblastoma. Additionally, a study showing that low socioeconomic status was associated with inferior survival in children with high-risk neuroblastoma. Our central hypothesis is that poverty and low socioeconomic environments are playing a role in the pathogenesis of neuroblastoma that surpasses the original incidence alone. And we also wanted to show that we could use an animal model to prospectively and ethically study the effects of stress. All our social experiments were based on our previously published neuroblastoma model that involved percutaneous injection with ultrasound guidance, and that allows us to see the tumor be injected directly, as well giving us a higher yield and a lower mortality. Our first study that we looked at was food insecurity, and we adopted this protocol from a previous study that involved placing a food insecure group with variable amount of food that they had access to, and we compared this to a control group that had open access to food at all times. Our initial results showed that we had a doubling or an increase in tumor size, almost 2 times the control group. Additionally, we found that corticosterone, adrenaline, and noradrenaline had almost 2 times the amount as well in the food insecure group compared to the food secure group. So we then took these pilot results and expanded it to different social stressors, specifically social isolation and social defeat. We know that, again, stemming from COVID-19, social isolation was being studied to determine the effects that it was having on the mental health of children, but less studies are known about what the effects on the pediatric cancers are. And so we know that in adults, additionally, they have been found to have increase in their central neuroendocrine signaling from the effects of social or chronic social isolation. So again, we used our animal model, our neuroblastoma model, after injecting the tumor and confirming that we had tumor growth after 4 weeks. We then induced our stress. One group was a normal caged group per standard guidelines, where the second group was isolated in individual cages. We found that, again, similar to our food insecurity group, that we had a doubling in tumor size in our social isolation group compared to our groups that were caged normally. And it's not pictured here. There was also an increase in the corticosterone, the adrenaline, and the noradrenaline levels as well. So then our last study was inspired by a group that did lung carcinoma in mice that were getting repetitive social defeat. And we found that they had increased amount of tumor growth and angiogenesis. And so we took another group, injected them with our neuroblastoma high-risk cells, and we also had a group of mice that were retired breeders, and we isolated them for 1 week so that they could establish their territory. The group that was designated to be stressed was then caged with the aggressor mice for 1 hour a day. For the first 5 minutes, they was allowed to have direct contact, but then the rest of the remaining time, they were separated by this clear plexiglass that you can see in this picture. And we did this each day for 15 days total. Unfortunately, we did not see the same results as our previous studies. There was no statistically significant difference between tumor volume in our corticosterone groups. And we are attributing this to the fact that we did a slight variation of the original protocol that had a longer period of stress, and we did a shorter period of stress, so that we had to repeat that study. Okay, so in conclusion, we were able to demonstrate that an animal model could be used as a method to study the social deprivation. We were able to find significant difference in the food insecurity group and our social isolation group, but we were not able to recreate those same situations with the social defeat group, but that study is undergoing new studies. Additionally, we are going to try different stressors such as learned helplessness, and even consider doing a stress prenatal model to help correlate with more human-like conditions. But we understand that we still need to understand the pathways more specific. So, thank you.
I may have missed it, but I have a question.
Sure.
Did you have a model where you had induced the stress or the food insecurity for a long period of time beforehand, and then as you were initiating the tumor injection, then reversed those things? Is there a way to impact the tumor growth and overcome the previous?
So that's like the next step of our projects. This was just a pilot study to see if we would even see a difference in the groups, and so that's the next thing that we're looking at, actually. Thank you. Yes.
Hi, this is Lisa Tracy from Duke. This is really impressive work.
Thank you.
And taking it into animal modeling, I think, is an incredible step here. I'm wondering if you've generated sort of survival curves or have some longer-term data on the mice.
No, we have not. Like I said, we were just trying to see if this was going to be a factor, but that is something that we can definitely consider in the future.
I'll look forward to following you.
Thank you.
Great work. Casper Wong from Toronto. I have 2 questions for you to speculate on, because I understand this is a pilot study. Can you speculate on what might happen if you tried to rescue these mice? So they've been food deprived, and then obviously we're looking for ways to intervene and improve outcome. What would happen if you were able to actually rescue them in terms of food deprivation, providing access, and then the other one, obviously, with the social deprivation, if you were to return them back to, socialization. And the second question is, there's a ton of investigation into the immune mediation of tumors and natural killer cells and all of that. You looked at a number of hormones, but did you look at any immune mediators, or can you speculate on what might be the result of some of that? Because obviously, it's hard to connect adrenaline to how the tumor might grow, but maybe something more closely aligned to tumor mediation and things like that. So great work, keep it up, and looking forward to seeing more in the future.
Thank you. So the second question is actually a preview of my next presentation, where we are looking at some of the immune landscape. But the first question, after we got these results, we were thinking, how can we show, I guess, along the lines of epigenetics or anything that we can change to make it reversible? So we're planning to start just looking at the cells, so looking at the tumor cells, treating them with different catecholamines and seeing if we can cause the same growth just at a cellular level. And then we'll start thinking about how if we can reverse it, or with not beta blockers, but like mTOR inhibitors and different things to kind of reverse the stress. So it's our next step. Thank you.
Hi, Liz Byerly from Alabama. Just a short comment about what you just last said. You may want to look back at some presentations that were done at some ASC meetings, because we've actually looked at treating the cells with stress hormones to see what they do in the dish, and it's pretty interesting. But my question to you is, so I was wondering if you have looked at actually starving the animals before you give them a tumor, because I think that's a little bit more of the situation that we're dealing with. So actually, once these kids get in the system, a lot of them, we can help them. And so my question is there something that's going on before they get a tumor that is affecting the whole issue? And I don't know if you've thought about that. Or
No, that's a good point. I know right now we're looking at just the food insecurity in general without tumor growth, and looking at stress hormones in that level. But we didn't think about inducing the stress prior to tumor injection. So that is something we can consider.
Congratulations, terrific work.
Thanks.
Our next speaker will be Dr. Doyle, who's presenting a comparison of in vivo tumor honing abilities of placental-derived and bone marrow-derived mesenchymal stromal cells in high-risk neuroblastoma.
Hi, everyone. I'm Kate Doyle. Thank you for the opportunity to present some of our work today. These are our disclosures. So to address the question of why we did this research and why we think it's important, as we've heard from some previous presentations, neuroblastoma is a common pediatric cancer and is the leading cause of cancer-related deaths in children under 5. There remains poor outcomes in high-risk disease despite rigorous multimodal treatments, and the side effects of these systemic cytotoxic treatments can result in life-altering morbidities and even death in these young patients. Therefore, there is a need for targeted therapies that are able to address both the heterogeneity of disease as well as mitigate some of these side effects of cytotoxic systemic therapies. Mesenchymal stromal cells, or MSCs, are a diverse group of cells that demonstrate multipotent differentiation capacity and can be isolated from various tissue sources. MSCs have been studied in oncology because they exhibit tumor migration or homing abilities, where they can travel directly to the tumor site. This has been utilized to engineer MSCs to carry cancer therapeutics directly to tumor sites, which allows for increased efficacy as well as decreasing collateral toxicity. This has been studied in multiple oncologic models, including some preliminary studies in neuroblastoma. Our lab specifically works with placental-derived mesenchymal stromal cells that are cultured in a neurogenic media and have demonstrated a unique neurotropism in a spinal cord injury model. While there hasn't been a lot of work utilizing PMSCs in oncology, there is some data that's similar to other types of MSCs. They exhibit similar homing abilities to tumors in vivo. We were curious if using these PMSCs with this unique neurotropism would demonstrate superior homing abilities in neurogenic tumor types, such as neuroblastoma. Therefore, we hypothesized that in comparison to bone marrow MSCs, PMSCs would demonstrate superior tumor homing abilities in neuroblastoma in an in vivo model. We chose bone marrow MSCs as a comparison because they are very widely studied in oncology and have also been studied and demonstrated homing in neuroblastoma tumors. This is our experimental design. We start by inducing neuroblastoma tumor growth in an orthotopic model. We monitor the tumors with ultrasound until they are about the size of the ipsilateral kidney, after which we perform MSC injection, and then we monitor homing using the in vivo imaging system, or IPSO, and subsequently perform necropsy at either 72 hours or 7 days, and histologic analysis to assess for the presence of MSCs in the tissue. We investigated 2 modes of MSC injection: direct intraperitoneal injection and intravenous injection via tail vein. We use a retroperitoneal surgical approach to induce neuroblastoma tumor growth, and then we monitor by ultrasound. We utilize the in vivo imaging system, or IPSO, to track our MSCs. MSCs are transduced with the luciferase gene, as well as green fluorescent protein, or GFP, which allows for imaging, a visualization on the IPSO as well as on IHC, respectively. At the time of imaging, each mouse is injected with luciferin, which then generates the bioluminescent signal that we can see as this multicolored signal shown on the mouse in the lower left side. So these are our results for the intraperitoneal injection of PMSCs. As we can see, the MSC signal starts out more diffusely initially, and then as imaging progresses, becomes more localized. On ex vivo imaging at time of necropsy, we see that there is an IVUS signal present in the adrenal mass and exclusively the adrenal mass. These are the intraperitoneal injection MSC homing results for our bone marrow mice, and similar to what we saw with the PMSCs, we see a more diffuse signal initially, which becomes more localized with time. And again, on ex vivo imaging at both of our necropsy time points, we saw a signal in the adrenal mass and only the adrenal mass. We performed immunohistochemistry staining for GFP, which confirmed the presence of MSCs in the tissue for both MSC types. And then we used the radiance measurement, which is given out by the IVUS machine, to quantitatively compare the homing of the PMSCs to the bone marrow MSCs. We did this by looking at the ex vivo radiance measurements given out by the machine on the adrenal masses, and we looked at them at 72 hours, which is shown here. On the y-axis, we see the log of the radiance measurement, and on the x-axis, we see MSC type. As we can see, there was no significant difference in radiance measurements of the adrenal masses at 72 hours. And this is the same figure, but for the mice that underwent necropsy at 7 days. And once again, we saw no significant difference in the radiance measurements of adrenal masses at 7 days. We also were interested in investigating intravenous injection of MSCs. Previous literature has suggested that when MSCs are injected intravenously, they tend to migrate to the lungs and don't really tend to live much longer than 24 hours. So we were curious if this would be a feasible homing mechanism of delivery. So we looked at a couple mice and injected MSCs via tail vein. As we can see here at time 0, we do see an IVUS signal. However, after time 0, we actually saw no signal in any of the mice, so we elected to perform necropsy at 72 hours for all of these mice in order to maximize the chance that we would be able to see the MSC signal on ex vivo imaging. And as we can see here, similar to what the literature reports, we saw that there was MSC signal exclusively in the lungs, and no signal was seen in any of our adrenal masses. So in conclusion, we saw that both types of MSCs migrated to the tumor when they were injected intraperitoneally. We did not see any difference in the homing abilities of PMSCs compared to bone marrow MSCs. And the intraperitoneal injection method appears to be more efficacious than intravenous injections. So moving forward, we're interested in investigating the pharmacokinetics of MSC homing by tracking them in real time with photoacoustic ultrasound. We're also investigating multiple methods of utilizing these MSCs as a drug delivery vehicle for microRNA therapies, which we'll hear a little bit more about later. I'd like to thank my lab group for all of our work, and I'll be happy to take any questions.
Hey, that's great work. I really like it. I like the homing aspect of it. Radiance for quantifying these things gets really tricky because depending on what the saturation level is of your settings and how much you're getting signal washed out within your masses, and that's reflected in sort of your error bars and everything. Have you done like IHC or dissociation and flow to really try to see how many actual cells are in there compared to what you injected? Because you may actually see differences there that IVUS really has trouble showing.
Yes, that's an excellent point. There's a lot of limitations to using the IVUS, and we did do IHC. We had a lot of trouble with actually finding the MSCs because they're big tumors and it's kind of like looking for a needle in a haystack, but we were able to successfully find them for both MSC types. So we did confirm that they did migrate there. Obviously, that's a more qualitative way of looking at that. In terms of, we discussed both flow cytometry and then utilizing PCR also to quantify the amount of MSCs that were there. For these initial studies, we were kind of leaning more towards just a kind of proof of concept pilot mentality, and ultimately kind of ran into the same thing of we would need to use the whole tumor, digest the entire tumor tissue, which based on all the studies that we wanted to do, we didn't want to sacrifice the entire tumor because again, if we just use a portion of the tumor and do PCR on that, we have no idea if the MSCs are going to be there. So we have discussed doing that with future tumors, but I think we now have a collaborator in our lab that is an expert in photoacoustic ultrasound, and so we're kind of starting with that because we're tagging them with cold nanoparticles so that we can actually watch where the MSCs are going and hopefully kind of see how we can get a quantification via that. But we certainly have discussed both flow and PCR to get a better quantitative comparison.
Thanks.
Hey, great talk. Marcus Malk from Pittsburgh. Very exciting stuff. Any way you can target specifically to a tumor obviously is going to be advantageous. I have a comment and a couple questions. The comment is sort of similar to the last point. I mean, you certainly can quantify regions of interest using the IVUS, so that may be one way that you can quantify. You could also potentially tag it to a radiometal or something like that and then do a gamma biodistribution. That would help in terms of really quantifying the amounts that are in various other tissues. The questions, what advantage do you think that this technique would have over using GD2 if you're planning to deliver medications or something else? As we know, that obviously targets well to neuroblastoma. And then my next question is, which cell line did you use for this? Have you tried it in multiple cell lines? Do you get a sense that it's going to be fairly ubiquitous across neuroblastoma cell lines and hopefully in patients in the future?
Yes. Thank you for the comment. That's a great suggestion. We'll definitely keep that in mind and talk about how we can incorporate that. And then in terms of the second question, we haven't specifically looked at using cell markers to do homing. We just were interested in investigating these PMSCs that we have because we thought they were a unique utility in our lab. And so that's why we thought there might be an advantage of superior homing because they have demonstrated this neuroaffinity, neurotropism in our spinal cord and spina bifida model in our lab. So that was kind of the idea behind that. We've looked at, so the cell line that we used for this, we used one high-risk neuroblastoma cell line, NV1643. We actually are doing some studies right now where we're using other cell lines as well as kind of an expansion on that because, of course, neuroblastoma is so heterogeneous, we don't know. And then the other element of that cell line is then the MSC cell lines as well. So we have a lot of PMSCs and bone marrow MSC cell lines. One of the ways that we looked at that previously in order to kind of narrow down which cell line to choose was one, comparing the neurotropism effects that we've done in the studies for our spina bifida model and selecting both high neurotropism ones and low neurotropism ones and comparing the effects of that. And then we also were concerned of the proliferative effects that MSCs could have on the tumor. So we did some studies last year looking at the multiple bone marrow cell lines, multiple PMSC cell lines to see which ones had lower proliferative effects on MSCs. And actually moving forward, we're planning to use one of our bone marrow cell lines that had the lowest effects because we did see more proliferation from the PMSCs, not really unexpectedly given that we know that they have this neurotropic effect as well. So,
thanks, great work.
Thank you.
One quick one because my colleague stole one of my questions. I'm Erica Newman from Mott, University of Michigan. This is so exciting. You did an amazing job. This is amazing work, so thank you.
Thank you.
I will shorten my question and then just a quick comment. Since our lab is very interested in sort of how the mice are stressed now because of the work we're doing with social deprivation, you can see the adrenal gland via ultrasound, and I see that you guys are being very technically savvy with the ultrasound machine. So have you considered just doing your injections via subcutaneous approach by not opening them, not operating on them in the very beginning? Because what we found is you can do probably up to 12 an hour. It's very stress-free on the animals, and you don't have to open them, operate on them, do your injections, and then close them and have them recover. It's just a real quick injection. So.
Like a subcutaneous tumor tissue or percutaneous into the adrenal?
Percutaneous.
Yes.
Right
so,
into the adrenal gland. Yeah.
We're actually really excited. We didn't have an ultrasound that had resolution high enough. We experimented with some percutaneous injections early, but ultimately weren't getting reliable results. But our same collaborator who has the photoacoustic ultrasound experience has a much better ultrasound technology. So we're actually right now also moving towards a percutaneous model going forward. So we're really excited about that because we think it's going to drastically change our lab.
Anything I can do to help you, let me know.
Thank you.
Nice job.
Thank you so much.
Thank you very much. All right, and now for a repeat performance. Dr. Kiona Williams will be presenting a characterization of neuroblastoma circulating tumor cells, reveal insights into disease progression and immune landscape.
And thank you for this opportunity to present. No disclosures. Circulating tumor cells, or CTCs, we know they're shed from the primary tumor and shed into the bloodstream. Excuse me, and that they can provide information about the metastatic potential of a tumor. Excuse me. Yet the clinical significance of these cells, and as well as the technology to actually isolate them, but their high yield and specificity is still being developed. Additionally, the findings specific for circulating tumor cells are limited in the pediatric cancers. So we did this experiment to, one, look at our novel technique, which involves microfluidic high-throughput Lambert device. And we wanted to look at specifically the context of CTCs and high-risk neuroblastoma. So we're hoping to present our validity and the clinical importance, as well as show the emergence of the immunocells as they are emerging in immunotherapy. So as previously mentioned, our technique would allow us to get a complete cell profile without being biomarker dependent. And we believe that's what allows us to have a better cell integrity that leads to a better analysis with our RNA sequencing. Additionally, clinically, this will be minimally invasive as we can get it from a peripheral blood sample that doesn't require tissue biopsy, and it will allow for easier clinical monitoring during clinical surveillance. So our aim was to isolate neuroblastoma circulating tumor cells. We then would perform single-cell RNA sequencing to identify the different characterization of the immune cells, and we want to look at specifically the tumor microenvironment. So first, for our isolation, we use our Lambert device, and after we isolate the circulating tumor cells, we then perform enumeration, which involves quantifying and identifying the different cells based off of the biomarkers dependence in immunostaining. We then perform our single-cell RNA sequencing, as mentioned. So this is a simplified illustration of our device, but essentially we inject our sample into our Lambert device, and using a combination of inertial and drag forces, we're able to push the particles along, and at different loops and curves along that pathway, we're able to isolate or separate off the circulating tumor cells because they have a size difference between the white blood cell counts. And then once we are able to isolate them off, we then collect them in this outlet system, and then they undergo sequencing. We then, for enumeration, we stain them for different cell markers, specifically DAPI and FOX2B and CD45. This is to kind of help, like I said, validate and identify that we actually have circulating tumor cells, and we have our merged immunostaining image here at the bottom to show how we were able to quantify them. We were able to get about 150 to 300 circulating tumor cells in 1 milliliter blood. This is compared to other techniques that have required 7.5 milliliters of blood, which has more of an impact in our pediatric population. So looking at our sequencing data, we were able to identify different immune clusters from our CTC sample, and I've highlighted them here in red. We're still kind of deducing what exactly this information would mean specific for our patients, but there are some studies that are showing that plasmacytoid dendritic cells might be inducing some of the lytic functions of natural killer cells. And they also might be able to induce more anti-GD2 mediated cell death in tumors that have low or moderate amounts of those receptors on board. And as we know, dinutuximab is one of the drugs that could potentially be used in this situation. So we're still working out through those results. Additionally, we did a KEGG analysis, which looks at the different genetic components, and it looks at on a higher cell level of how those things are interrelated. We did multiple pathways, but the ones that I will highlight here specifically is just that our immune-specific pathways. We saw that there was an enrichment in along the cancer pathways, as well as enrichment in our natural killer-mediated cytotoxicity pathway, again, validating that we were able to isolate neuroblastoma cells and kind of highlight some of the potential that we could use for immunotherapy. Excuse me. Oh, okay. So in summary, we were able to isolate these neuroblastoma cells using our Lambert-Feed device. We felt like with a high specificity that allowed better characterization of the immune cells and allowed better patient-specific information with our RNA sequencing analysis. Future studies, though, will need to look at specific diagnostic and therapeutic targets that we can identify. Thank you for your time. Acknowledgements.
I'll ask a question. Congratulations. This is very interesting work, and I think the role of circulating tumor cells is certainly being played out in a lot of different arenas. As we think about the heterogeneity of neuroblastoma, and you touched on this briefly, but I'm curious to hear your thoughts more. How do you see this playing out in a translational fashion for our patients, and what do you think the clinical application could be?
So as long as the clinical application of this, so I think one of the tricky parts that we are realizing as we collect more samples is that it's individual to each patient. So we're having to figure out how to find a pattern along those patient samples because it's such differences. But the idea is to collect it over different periods of time. So our initial study was looking at collecting them pre-surgical, so after they had an induction chemotherapy. And so we want to start looking at what our cell line looks like before surgery, before any chemotherapy, and after kind of surgical stuff. So we're still working on the different time points of when to get it so we can figure out how to incorporate this. Thank you.
Thanks. David off Memphis, very nice presentation. Can you speak to the differences between your approach looking at circulating tumor cells versus the cell-free or circulating tumor DNA, which actually has already made its way into current COG clinical trials for neuroblastoma? Why do you think your approach or target is better?
Right. So I think the biggest thing is that we feel like we're able to get a higher volume or a higher number of circulating tumor cells. And then by doing single-cell RNA sequencing, we're able to get like a complete I think a complete set of genetic information that we think allows us to kind of just explore the different, facets of that tumor population. So that's I think one of our benefits is that we're just getting like as complete of a cell profile as possible.
Thank you. Thanks. The next speaker will be presenting on co-localization of tumor-associated macrophages and regulatory T cells creates immunoregulatory niches in human hepatoblastoma. Please welcome Dr. Kotemraju to the stand.
Good morning, everyone. My name is Mahadi Kotemraju, and I am an undergraduate and rising sophomore at Boston University and a student researcher at the University of California, San Francisco. I began researching at UCSF when I was a senior in high school, and the study is titled Tumor-Associated Macrophages and Regulatory T Cells May Create Immunoregulatory Niches in Human Hepatoblastoma. So as many of you are aware, hepatoblastoma is the #1 cause for primary liver tumors in infants and children. And while the prognosis is good and therapies have progressed enough that prognosis is good for those patients with low-risk tumors, unfortunately, those with high-risk tumors still face a high morbidity rate. So a characteristic of this cancer, which often complicates treatment outcomes, is the high levels of tissue heterogeneity that are displayed by the cancer. So within this 200 micron view, which is pretty microscopic, we can see about 3 to 4 different types of tissue just within this 200 micron view. So we previously investigated the immune cells that are found in the normal liver versus tumor tissue. And amongst those immune cells that are found in tumor tissue, we looked at the immune cells expressed at a higher frequency in tumor tissue. And of those, we looked at tumor-associated macrophages. So tumor-associated macrophages work with other cells to create an immunosuppressive environment. And those other cells that we looked at in this case were regulatory T cells, because they work to maintain this immunosuppressive environment. So our hypothesis was that tumor-associated macrophages, which I'll refer to as TAMs, and regulatory T cells, which I'll refer to as Tregs, co-localize in human hepatoblastoma. So we first began by collecting 9 tissue samples. So 1 tissue sample was collected from 9 separate pediatric hepatoblastoma surgeries. And we stained these 9 tissue samples with H&E in order to identify the areas of normal tissue and tumor. So here's a pretty clear boundary between the normal and tumor tissue of this patient. And right of this image is a more macroscopic view of what this boundary would look like. After staining with H&E, we stained with our 2 immune stains in order to identify our immune cell populations of interest. So we stained with CD68 in order to identify our TAMs. And at a microscopic view, we were able to identify our CD68 positive cells, which are our TAMs. And at a macroscopic view, we were able to identify our clusters of these cells. We did the same for FOXP3 stain, which stained for Tregs. At a microscopic view, we were able to see our FOXP3 positive cells, which are our Tregs. And at a macroscopic view, we're able to see our clusters. We then looked at the overlap between these 2 immune stains in order to compare the cell populations and their clustering to determine whether they were co-localizing. And here's a pretty clear example of some clear co-localization in this patient. So for reference, this is the same section of tissue, the same area of the same patient, just with the 2 stains side by side, which offers some pretty nice comparison. In order to streamline our data analysis after this, we set definitions. So we defined a cluster as at least 3 cells appearing within 80 microns of each other, and a cluster as a cluster of TAMs and Tregs appearing within 30 microns of each other. We then collected data on the number of cells per cluster, the size of the cluster, the number of clusters co-localizing, and confirmed the results of our analysis with pediatric liver pathologists and an expert in Treg stain. So based on our data, we were able to see that there are significant differences in the characteristics of these 2 immune cell clusters. So when looking at the average number of cells per cluster, we're able to see that TAM clusters, for lack of a better term, are denser and have a greater number of average cells per cluster compared to Treg clusters. The average distance between cells per cluster was also significant, but the results were more comparable. And the average size of the clusters was different, again, with TAM clusters being much larger and taking up much more space in the tumor compared to Treg clusters. When looking at our co-cluster data, we were able to determine that 36% of Treg clusters co-localized with TAM clusters. So out of our 167 Treg clusters, we had 60 co-localized with the TAM cluster and 107 not co-localized. Out of our 120 TAM clusters, 60 co-localized with the Treg cluster and 60 did not. So it's notable that there were no clusters or co-clusters in normal liver. We only found clusters in tumor tissue. When looking at this data in terms of percentage, we can see that a higher percentage of TAM clusters co-localized with the Treg cluster when comparing Treg clusters to TAM. So from this data, we can conclude that Tregs and TAMs form clusters in hepatoblastoma tumors, but do not form clusters in normal liver, and that TAMs and Tregs co-localize in hepatoblastoma tumor, suggesting that they form immunosuppressive niches. A couple things to note about this study. Almost all of the patients had received chemotherapy prior to tissue resection, which is significant because, amongst other things, chemotherapy can affect the behavior of these immune cells, and a direct interaction between TAMs and Tregs has not yet been established. When looking at future directions, I'd want to take this study forward in terms of looking at the correlation between co-clusters and patient outcomes. So looking at the location of these clusters, the presence of these clusters, whether that indicates good or bad prognosis for the patient. And the study would not have been possible without the incredible support structure behind me. I'd like to thank Doctors Cho and Cohen from UCSF for their help in the verification of our cluster data. I'd like to thank my PI, Dr. Amar Nijigal, for his constant encouragement and support. And I'd like to thank the entire Nijigal lab for all of the learning opportunities and all of the feedback. Thank you.
I have a question. Could you explain, in your abstract, you talk about Tregs co-localizing with TAMs, but then you seem to differentiate that from TAMs that co-localize with Tregs. I don't understand the differentiation.
So I don't know if I can go back to the slide. It's probably, well, never mind, I guess I can. So essentially, we had a different number. Oh, I can. No, I can't. I'm so sorry. I'm so sorry. We had a different number of, there we go. We had a different number of clusters for our TAMs and our Tregs. So because there was a higher number of Tregs, there was 167 of them, and there was only 120 TAMs. When we looked at, so if I break this down in terms of TAMs, I think it's easier to understand. Out of our 120, 60 of them were found with another Treg cluster. So when you look at the tissue, you look at the two stains side by side. You switch over to the Treg stain for that same area of tissue, you find a Treg cluster. But if you were to go to, let's say, a Treg cluster and it's not a co-cluster, you look at the tissue, you won't find a TAM cluster in that area. I'm not sure if that answers your question. I feel like a visual.
I'm not sure either.
I'm sorry about that.
I'm not going to tie up everybody else's time with my ignorance.
I think if we don't have any other questions, we'll go on to our next speaker. You did a terrific job. We hope you'll think about pediatric surgery.
All right, so the next abstract is multi-tome sequencing reveals common transcriptional circuitry and gene expression patterns in high-risk neuroblastoma cells, regardless of MICN status, being presented by Dr. Richard Geyer.
All right, good morning, and thank you all for the opportunity to present this work here. We have no relevant disclosures to report. We've previously published that neuroblastoma cell lines without MICN amplification are heterogeneous, containing both immature proliferative cells marked by the CD49B antigen and quiescent cells that express neuronal marker genes and lack CD49B. However, MICN amplification is the most common cause of mutation in neuroblastoma and is seen in a large fraction of high-risk tumors. Our prior work was thus likely not relevant to many of the most aggressive lesions. We undertook this project to better understand the biological differences between tumors with and without MICN amplification. We hypothesized that cellular heterogeneity and transcriptional programs would differ in MICN-amplified neuroblastoma as compared to tumors lacking MICN amplification. To address these questions, we used 10x Genomics' commercially available multi-omic sequencing kits to simultaneously assay gene expression and chromatin structure in individual tumor cells from several neuroblastoma cell lines. Computational analysis of gene expression and chromatin accessibility was used to identify key regulatory genes in each cell line. We then compared expression of these key genes across cells with and without MICN amplification. We used 6 cell lines for this study, 3 each with MICN amplification and without MICN amplification. I want to start by briefly reviewing a couple terms that are important for this talk. First, ATAC, which is an abbreviation of Assay for Transposase Accessible Chromatin. When histones are exposed to transposases, DNA that's coiled about histones and heterochromatin is protected from digestion. The ATAC assay takes advantage of this feature and utilizes transposases to digest open chromatin into short fragments, while sparing heterochromatin genomic regions. The fragments generated by the transposases are then isolated by size selection and sequenced on next-generation platforms. The sequencing reads are aligned to a reference genome, thus giving a genome-wide, unbiased snapshot of chromatin accessibility in a cell. ATAC data has become widely used to study chromatin state and gene regulation, and it's particularly useful when combined with gene expression data. Transposase sensitivity, as measured by the ATAC assay, correlates strongly with gene expression and activity of critical DNA regulatory elements. Another concept that has recently emerged from combined analysis of ATAC data and gene expression is the domain of regulatory chromatin, or a DORC. A DORC is a large chromatin region defined by numerous ATAC peaks that are strongly associated with a particular gene of interest. DORCs tend to overlap to a large extent with superenhancer regions and are usually associated with genes that play a key role in determining cell phenotype. ATAC signal within DORCs is strongly correlated with gene expression of the DORC-associated gene, although signal at DORCs may precede gene expression. We first examined heterogeneity of neuronal gene expression in our cell lines without MICAN amplification. As expected, based on our previous data, we found an inverse correlation between expression of ITGA2, which is the gene that encodes for the CD49B antigen, and expression of neuronal markers, such as SNAP25. In contrast, MICAN-amplified cells show more uniform expression of neuronal markers. SNAP25, FOX2B, and SYP, all of which are expressed in mature neurons in human and mammals more broadly, are all expressed fairly broadly in MICAN-amplified cells. Interestingly, ITGA2 transcripts are detectable at low levels in a subset of MICAN-amplified neuroblastoma cells. However, in contrast to cells lacking MICAN amplification, there is no clear association or pattern with relation of ITGA2 gene expression and neuronal gene expression patterns. We then turned our association to chromatin structure and analyzed DORCs. We identified 628 DORC-associated genes in our 6 neuroblastoma cell lines, most of which were associated with a DORC only in one cell line. A few, however, were active across multiple cell lines. When we examined transcription abundance of these DORC-associated genes, we found that many were strongly associated with a particular cell line. However, especially near the midpoint of the range of gene expression, there were numerous genes with detectable transcripts across all cell lines. We focused on several of these broadly expressed genes for further analysis, choosing to focus on genes with known cancer associations. TNC and MyCal2 are both associated with tumor growth and progression in numerous cancer types, including breast adenocarcinoma, head and neck squamous cell cancer, prostate cancer, and gliomas. TNC encodes for the tenascin C extracellular matrix protein, while MyCal2 encodes for an eponymous microtubule-associated protein. Our analysis identified a TNC-associated DORC in the SH-SY5Y cell line, and the MyCal2-associated DORCs were identified in all 3 of the non-MIC-amplified neuroblastoma cell lines we analyzed. Interestingly, chromatin accessibility at TNC and MyCal2 DORCs seems to correlate strongly with ITGA2 RNA abundance. Similarly, transcript abundance of MyCal2 and TNC correlates with ITGA2 transcript abundance in all 3 of these cell lines. These data suggest that TNC and MyCal2 may be important drivers of immature proliferative cell state in neuroblastomas without MICAN amplification. Despite not being associated with a DORC in MICAN-amplified cell lines, both TNC and MyCal2 transcripts are detectable in MICAN-amplified cells. TNC has a low level of expression, but MyCal2 is highly expressed in at least some of the cells in all 3 cell lines we analyzed. This suggests that MyCal2 might be an important driver of the tumor phenotype regardless of MICAN status. While TNC is expressed at low levels in MICAN-amplified cells, it is secreted ECM protein whose abundance at the transcript level may not correlate with protein abundance in the extracellular environment, and so it may also be an interesting target for further study. ID1 is a DORC-associated gene identified in all 3 MICAN-amplified cell lines, and all 3 of these cell lines express high levels of the RNA. ID1 encodes for an eponymous DNA binding protein that is highly expressed in several cancers, where it is uniformly associated with an aggressive cell phenotype, an immature cell state, and poor patient outcomes. This includes cervical cancer, colon cancer, and glioblastoma multiforme. During normal development, ID1 is expressed in immature cells of numerous lineages, where it's believed to impair expression of mature genes and prevent premature cell differentiation. An ID1 associated DORC was also seen in the non-MICAN amplified SKN-AS cell line, and ID1 transcripts are found in all 3 of these non-amplified lines. There does appear to be some association between expression of ID1 RNA and ITGA2 RNA. These data indicate that ID1 may be a key regulator of transcriptional circuitry in neuroblastoma cells, possibly granting immature aggressive features to tumor cells. So in conclusion, joint measurement of ATAC signal and gene expression in individual cells has demonstrated that MYC-N amplified cells have less heterogeneous neuronal gene expression patterns than non-amplified cells. MYC-N amplification thus lends aggressive features to neuroblastoma cells, despite expression of genes that are typically associated with a mature quiescent state. This is a clear divergence from MYC-N non-amplified tumor cell lines, in which immature cells without neuronal markers are proliferative, while cells expressing neuronal genes are quiescent. Despite these differences in neuronal gene expression patterns, we identified several cancer-associated genes that are expressed widely in all neuroblastoma cell lines we sequenced. ID1 may be a particularly good candidate for unraveling transcriptional circuitries important to high-risk neuroblastomas. Thank you all very much for your time. I apologize for running a few seconds over, and I'd be happy to take any questions if there's any time left.
David off Memphis, thanks very much, Rich, for a fantastic presentation, a very sophisticated methodologies, very, very well explained. I have a couple of questions. One is, were you surprised by the homogeneous nature of the neuronal markers in the MYC-N amplified cell lines, given that, okay, they all were NMYC amplified, but there are a number of other segmental chromosomal aberrations and so on that are going to be different among even MYC-N amplified cell lines. And I'll let you answer that, and I'll
yeah, the answer is yes, I was. Given the heterogeneity we see in non-amplified cell lines, I expect we'd see something similar with MYC-N amplification, but at least the 3 lines we looked at, and we only looked at 3, so maybe I just got lucky or unlucky depending on how you look at it, but if we looked at 20 cell lines, we wouldn't have seen the same thing, but at least in these 3, I was surprised by that effect.
And thanks. So the second question is, there's a lot of talk about cell state in neuroblastoma, so mesenchymal state, adrenergic state. Does that distinction, how does that relate to your observations?
Yeah, I think in the MYC-N non-amplified lines, based on our previous work that we show here, the adrenergic state is I think associated with the expression of the neuronal genes, or the more quiescent cells, and then the more mesenchymal state, is the term people applied to it, I think really reflects the ITGA2 positive cells. That's something we published previously on. In the MYC-N amplified cells, I'm not sure, we haven't gone to the level of analysis to be able to speak to those yet, but the fact that the neuronal marker genes are so broadly expressed, to me indicates that might not be just a clear-cut difference with MYC-N amplification.
Thanks, very nice.
So I would assume the next stage is to take this to tissue samples?
Yeah, certainly. Looking at patient tumors, tissue samples, and animal models, I think would be the next step. Also, whenever you're looking at sequencing data, it's a great hypothesis generating tool, but I don't think it settles any questions. I think you have to look at protein activity and what's going on at other levels of phenotype regulation before you can really say what's happening. Thank you all very much.
Thanks. Now we welcome Dr. Doyle back to the podium to present novel bioengineered microRNA therapy for high-risk neuroblastoma.
Thank you all for the opportunity to present some more of our work today. These are our same disclosures. And we've heard a lot about neuroblastoma. We know this is a common pediatric cancer with poor survival and high-risk disease, and high risk of recurrence and high-risk disease as well. That leads to even worse outcomes. Therefore, there is a high need for novel and targeted tumor-specific therapies to improve these outcomes. MicroRNAs, or miRs, are small single-stranded RNA molecules that regulate gene expression. miRs can be oncogenic or tumor-suppressing, and dysregulation of miRs can lead to tumorigenesis. miRs have been studied as therapeutics in multiple oncologic models, and have been shown to have therapeutic effects with restoration of dysregulated miRs. miRs have also been shown to work synergistically with systemic therapies to improve therapeutic effects as well as decrease treatment resistance. We work in collaboration with a lab that has shown great efficacy of 2 specific miRs, miR34A and miR124, in a lung cancer model. And we were curious if these therapeutic effects of these miRs could be translated to neuroblastoma. So both MIRS 124 and MIR 34A have been studied in the context of neuroblastoma and are tumor-suppressing MIRS that have been shown to be dysregulated in various tumors. MIR 34A specifically targets MYC-N as well as multiple other transcription factors and is known to play a role in multiple cell cycle progression as well as apoptosis pathways and epithelial to mesenchymal transition. MIR 124, on the other hand, primarily plays a role in neuroblastoma cell differentiation. It is known to be overexpressed in neuroblastoma cells that are treated with retinoic acid, as well as regulates multiple genes that play a role in neuroblastoma differentiation. So for our in vitro studies, we started with 3 high-risk neuroblastoma cell lines that were seeded on a 96-well plate at a concentration of 12,000 cells per well. We then added 25 nanomolars of either microRNA 34A or microRNA 124, along with the transfection agent, lipofectamine 3000, were added after 24 hours of seeding of the cells. They underwent an incubation period of 72 hours, after which we performed MTT cell viability assays and real-time quantitative PCR to assess for MIR levels within the cells. So here we see the results from our cell viability assays. The gray column represents neuroblastoma cells alone. The black column represents neuroblastoma cells that were co-cultured with the transfection agent lipofectamine 3000 only. Blue represents neuroblastoma cells co-cultured with or transfected with MIR 34A, and red represents neuroblastoma cells transfected with MIR 124. As we can see, for all 3 cell lines, MIR 124 had a significant decreased cell growth compared to the neuroblastoma cells alone. In cell lines SKNSH and BE2C, we also saw significant effects from MIR 34A on cell viability when comparing to untreated neuroblastoma cells alone. Another thing we were concerned about was the transfection efficiency of our transfection agent lipofectamine 3000. We were concerned about, based on looking at the literature with these cell lines, the transfection efficiency ranges from about 20 to 80%, which certainly could limit the ability of the microRNAs to show a therapeutic effect if they're not effectively getting into the cells and then being produced within the cells. So therefore, we performed real-time quantitative PCR to assess for the microRNA levels in the cells that were transfected compared to 2 controls. We used neuroblastoma cell lines that were treated with a non-specific microRNA, as well as cells treated just with the vehicle. As we can see here, the lines shown in red represent the cells that are transfected with microRNA 124, and we saw that there was a significantly increased production of microRNA 124 in the cells that were transfected compared to the controls, confirming that microRNA 124 is successfully transfected and the cells are producing it. And then we've completed this PCR for 2 of our 3 cell lines for microRNA 34A. And here we saw a little bit more variable results, but we did see that there was increased production of microRNA 34A in the transfected cells. This difference was only significant in SI5Y compared to BE2C, but we did see increase in both. So in conclusion, we found that both microRNAs 124 and 34A showed some efficacy in reducing cell viability in multiple high-risk neuroblastoma cell lines. However, 124 appears to have a greater efficacy than 34A. And then we know based on the literature and how these microRNAs function that a cell viability assay may not be the only or the best way to assess the effects that these microRNAs are having on the cells. And so we may need to look at other pathways to see the treatment effects of these microRNAs. So moving forward, we're planning on looking at functional assays of specific genes, as well as markers of differentiation to give a better understanding of the mechanism of how these microRNAs are exerting their therapeutic effects. We're also interested in pursuing studies of microRNA combination therapy with systemic cytotoxic chemotherapies, as well as retinoic acid. And we're moving this into our in vivo model, both the heterotopic and orthotopic. And then ultimately, our goal is to combine this with the previous research that we presented, utilizing MSCs to directly deliver microRNA therapies directly to tumor sites. Thank you to my lab team, and I'd be happy to take any questions.
Hi, thank you very much for a nice presentation. My name's Tammy, I'm from Nemours. I was very interested in your early slide looking at your cell viability and you're looking at your lipofectamine. alone, vehicle alone.
Mm-hmm.
Can you talk why you saw a difference in cell viability between your control and your vehicle alone?
Yes, that's a really important point. So we know that the vehicle has toxicity to the cells, and so we wanted to look at that. We actually standardized to the lipofectamine control to 100, knowing that there's going to be some cytotoxic effects of that reagent. It's also, it's a little challenging too because we use different transfection agents when we go to the in vivo model. So we're kind of working on also doing studies with the other transfection agent that we'd use in the in vivo model in order to get a better understanding of the effects of that.
So, of course, gene therapy for correction of cancer abnormalities is a challenge because you have to presumably fix that in all of the cancer cells, and so you have to have a great sort of targeting mechanism. So I wonder your thoughts on that. So you mentioned your MSCs as a delivery vehicle, but how does the microRNA get from the MSC into the cell, into the nucleus of the target cancer cells? So I wonder if you could talk a little more about that.
Sure. So with our collaborators who have done this in lung cancer, they've used the lipid nanoparticle, lipopolplex, something, to get their microRNAs into the cells. So that's actually where we're starting to. We're looking at using lipid nanoparticles that are anchored to the surface of the MSCs, and then when they're delivered, we're hoping that they will diffuse into the cancer cells and get in that way. But we also have investigating like a pH release system at the MSCs, at the cancer cells, to release the lipid nanoparticles there, and then again hoping that will translate to neuroblastoma cells the way that they have had success in the lung cancer model with our collaborators. But we're actively working on those in vitro studies to see.
Did you try combining the two mRNAs?
We have not yet, but our collaborators have also done that and showed improved efficacy when they use them in combination, so we're also interested in doing that as well. Thank you very much.
Thank you, nice job.
Thank you.
Next abstract is the role of YAP1 on hepatoblastoma vascular invasion, being presented by Dr. Andy Espinoza.
Thank you very much for the opportunity to present today. We have no disclosures. I'd like to start the talk by addressing the two questions of why we did the research and why it's important. And our lab primarily focuses on studying hepatoblastoma because it's the most common liver malignancy in children. And as you guys heard earlier, while patients with low-risk disease have survival rates that are greater than 90%, patients that have found to have vascular invasion have had reports of lower survival. In 2017, Dr. Shi published a study that focused on patients with vascular invasions. And as you can see here, those with vascular invasion, is shown in red, had about a 30% lower survival in patients that had similar characteristics, but without vascular invasion. But despite it being well understood that vascular invasion is a poor prognostic factor, little is known about the biological drivers, which was the aim of our study. To be able to start to do this, we use a technique known as imaging mass cytometry, where we use fluorescently tagged antibodies to simultaneously detect several markers of high-risk hepatoblastoma, which then allowed us to do single-cell comparison of these samples. We started by identifying 6 different patients that had vascular invasion, or VI, that did not receive any neoadjuvant chemotherapy, and then did a thorough validation of a 23 high-risk hepatoblastoma marker panel. As you can see here, these are the results of the imaging mass cytometry. We could simultaneously detect these different high-risk markers. And then there were several candidates that had upregulation as the tumor cells invaded into the vasculature. But of these, the one that most consistently upregulated was YAP1. When we validated this on the remaining samples, as you can see here, the cells in green are hepatoblastoma. As they invade into the vasculature, we could see upregulation of YAP1 as shown in red. When we quantified this, there was at least a 20-fold higher expression in the cells within the blood vessel compared to outside. And then to further validate this, we performed IHC staining of YAP1 on paired patients' primary tumors and metastatic lesions. and we're able to find an upregulation of YAP1 in the metastatic tumors. When we quantified this, there was about a 2-fold higher expression of YAP1 on the metastatic tumors compared to the primary. And given that we felt we had thoroughly identified this to be a specific target of invasion and metastasis, we sought to evaluate the role in vivo. For the first experiment, we used our non-metastatic cell-derived mouse model utilizing HepG2 cells that don't have the propensity to grow lung mets. We genetically manipulated these cells to have upregulation of YAP1, validating this on Western blots, and then injecting orthotopically into the mice. For the study, we had 8 mice into each group, 8 with parental HepG2 tumors in the liver and 8 tumors that had YAP overexpression. And we simply monitored the phenotype of these different mice. We didn't treat them with any drugs. We performed weekly MRIs and then euthanized them once the tumor volume reached greater than 1 centimeter cubed. This is the tumor progression of the 2 different groups. As you can see here, the YAP overexpressing tumors in black and the parental HepG2 tumors in red were very similar in growth. But what was interesting is when we sectioned the lungs of these mice and stained for GPC3, a marker for hepatoblastoma, we found very few lung deposits of tumors in the parental tumor-bearing mice, but then the YAP1 overexpressing mice, as you can see here in the red arrows, had several deposits of hepatoblastoma. When we quantified this, there was about a 5-fold higher expression in the YAP1 overexpressing tumor-bearing mice. Given that we felt we had found that overexpressing YAP1 would increase metastatic lesions, we sought next to validate this by seeing what would happen if we inhibited YAP1 in vivo. We used a chemotherapy drug known as CA3, which inhibits YAP1 activation and also decreases its expression. Started by first similarly validating that it actually decreased expression in orthotopic models as shown here in Western blots. For this study, we used the metastatic patient-derived xenograft using a technique that our group has previously published and had 2 different groups with the metastatic PDX, 3 that received the CA3 YAP1 inhibitor and then 3 that received the vehicle of the drug. And similar to the previous study, we had weekly MRIs that monitored the tumor growth and then euthanized them once the tumor volume reached greater than 1 centimeter cubed. Similar to the previous experiment, the tumor growth was fairly similar between the 2 different groups throughout the follow-up. But when sectioning the lungs once again, the CA3 group had very few lung deposits, as seen here in the black arrow. But in comparison, the vehicle-treated mice had several lung deposits of hepatoblastoma, as shown in the red arrows. When quantifying this, while not statistically significant, there was about a 1.5-fold higher expression in the vehicle-treated mice compared to the CA3-treated mice. So in conclusion, we showed that YAP1 increases in expression as hepatoblastoma cells invade into the vasculature and metastasize. Using our mouse models, we were able to show that increasing YAP1 of the tumors increases metastatic lesions, and while inhibiting YAP1 in vivo seems to decrease the amount of metastatic burden. And lastly, it appears that using experiments, we've shown that YAP1 does play a biological role in metastasis and vascular invasion that appears to be critical. I'd like to thank my mentor, Dr. Sanjeev Vasudevan, our lab, our funding sources, and our collaborators for all their support. And I'm happy to take any questions. Thank you.
Did you try other methods of inhibiting the YAP1 efficacy?
Yeah, great question. So we primarily used the CA3 inhibitor because when we had tried other inhibitors in vitro, it seemed that it wasn't as effective as other ones. So that's why we decided to use that specific inhibitor. Now, for further studies for more validation, I think using other methods of inhibiting YAP1 probably would make it more solid.
Joe Fusco from Nashville. So my question is similar to that. So that Hippo YAP1 pathway seems to be very diverse. So if you're able to decrease expression of that YAP1, do you think that you would prevent that metastasis?
I think that it would likely decrease the metastatic burden. I don't think that the YAP1 pathway is going to be the only thing that's leading to invasion and metastatic disease. I think it's a little bit more complex than that. But I do believe that if we were able to inhibit it using other inhibitors or manipulate it, I do believe that it would at least decrease some of the metastatic burden.
And my second question is for head and neck cancers. YAP1 is really important, but it's really the nuclear localization of YAP1. Did you look at the localization where it is?
Yeah, absolutely. That's another really good point. So with the imaging mass cytometry, we were able to co-localize it to the nucleus to confirm that it is upregulated. That's correct. And then the C3 inhibitor does play a role in not only decreasing the expression, but inhibiting its nuclear localization.
Great, thank you.
Yeah.
Is that it? Yeah. All right. Well, that concludes our presentation this morning.